We will now begin our discussion of aerobic respiration, first with an overview, and then after that we'll go into more detail. Now, aerobic respiration is common to prokaryotes and eukaryotes, but we will be focusing on eukaryotes. And looking at this picture, how do I know I'm dealing with a eukaryote? You might want to pause to figure out. Yes, indeed, because I have a mitochondrion here. So that's in eukaryotes. So first stage in the process, it's actually a metabolic pathway. So it's a series of chemical reactions that we call glycolysis. And glycolysis is not unique to aerobic respiration. Glycolysis is a very old metabolic pathway that actually evolved before aerobic respiration evolved. And glycolysis is used in other processes. So for example, when we talk about fermentation, that also includes glycolysis. So that will be stage one. Then we have a linking step where we're going to carry our intermediate substance. So we started with glucose and we made pyruvate with glycolysis, and then we're going to transport that pyruvate into the mitochondrion, where it's going to undergo the second stage. So stage two is also a metabolic pathway. It's a cycle, so we call it the citric acid cycle. And in that stage, we're going to produce ATP. I forgot to mention in glycolysis, we make some ATP already. In the citric acid cycle, we also make some ATP. And we also transfer electrons to those electron carriers that we have mentioned before, NADH and FADH2. Well, these are the reduced forms, right? So that's stage two. Then finally, stage three, which we call oxidative phosphorylation, which includes two processes, electron transport chain and chemiosmosis. This is when we're going to make most of our ATP by using the energy from those NADH and FADH2 molecules. Actually, I also forgot to mention that glycolysis not only makes ATP, but also makes NADH. So on this image, what we're looking at is the total amount of ATP that can be made by the process of aerobic respiration, starting with one glucose. So as you can see, we can make up to 38 ATPs, okay? And just in general, you can see that the glycolysis pathway makes some ATP, right? 2 ATPs. Here, it also makes some allows us to reduce some electron carriers to NADH, which can later on be used to make ATP in stage 3. The linking step also reduces some NAD plus to NADH, allowing us to make more ATP later on in stage 3. And same thing for the Krebs cycle. So it does produces some ATP directly, as well as reduces those electron carriers, allowing for more ATP to be made later on in stage 3. So as you can see here, there's two terms that we would like to define: substrate-level phosphorylation and oxidative phosphorylation. So substrate-level phosphorylation is perhaps more straightforward. What it is basically the transfer of one phosphate from a substrate to ADP to make ATP, to recharge our ATP battery with the help of an enzyme. While oxidative phosphorylation is a little bit more tricky to understand for now, but is the result of the use of NADH and FADH which are going to transfer their electrons to oxygen. It involves the electron transport chain and chemiosmosis. And in eukaryotes, all of this occurs in the mitochondrion. So these images probably don't make a lot of sense to you yet, but as you review the material after you've watched all the recordings, and then you can go back to your notes, and hopefully that will make some sense. Now we're going to be able to discuss those stages in a little bit more detail. As mentioned before, glycolysis is a metabolic pathway. It's a series of chemical reactions, 10 chemical reactions, and it's a very ancient pathway. It's found in nearly all living organisms. And it's also connected to many other pathways in our metabolic network, which we discussed in the previous week. So as you can see here, number 1 with the blue rectangle shows the different steps in glycolysis. So each circle is a substance, and the lines, the links between the circles are the chemical reactions. So as you can see, each substance is also connected to other parts of the network. The process of glycolysis does not require oxygen, and when we're talking about eukaryotes, it occurs in the cytosol. In very general terms, what we're doing is we're starting with one glucose molecule, which is oxidized to 2 pyruvate molecules. So glucose contains 6 carbons, and through all those steps, those chemical reactions, we're going to make 2 times pyruvate molecules, which contain 3 carbons each. In that process, we're also making 2 ATPs as well as 2 NADH. And NADH contains some energy that will be used later on to make more ATP. Just before we go on to the next slide, you can see here that the glycolysis stage, number 1, is connected to stage number 2 via the linking step. And stage number 2 is the citric acid cycle, which is also connected to many other parts of the metabolic network. And the last stage of oxidative phosphorylation is also part of our metabolic network. So we see again here glucose being oxidized through several steps to form 2 pyruvate molecules. Now, there are 2 phases in glycolysis. There's what we call the preparatory phase, where we're going to need to invest some ATP. 2 ATPs are needed for that phase. And then there's the payoff phase, which is going to result in production of 4 ATPs and 2 NADH. Now, we already expanded 2 ATPs, and we gained 4 ATPs, so we have a net of 2 ATPs that are gained plus 2 NADH. Okay, so we have an input of glucose. Our output includes 2 ATPs, 2 NADH, and 2 pyruvate. So we are now looking at the preparatory phase of glycolysis. And overall, what we're going to achieve is to oxidize a glucose molecule to form 2 glyceraldehyde 3-phosphate molecules. So our glucose molecules contain 6 carbons, and our glyceraldehyde 3-phosphate molecules contain 3 carbons. You can see here that we have 5 chemical reactions that take place. And you see here the glucose as our starting molecule. Here we have glyceraldehyde 3-phosphate, so that's 1. The other one is because we're going to convert dihydroxyacetone phosphate to another glyceraldehyde triphosphate, so we end up with 2 of those. You can see that we are actually going to invest 2 ATP molecules in that process, or use the energy from 2 ATP molecules in the first step and third step. And it's interesting to see also that these steps are the 2 irreversible steps. That is, you cannot go back. And these are important because they provide some control points in the metabolic pathway where you cannot go in the opposite direction. So let's look at our first step, the process of phosphorylation of the glucose molecule. So glucose, through the help of the enzyme hexokinase and through the input of 1 ATP, we're going to form glucose 6-phosphate. So as you can see, one of the phosphates was transferred to our glucose molecule, and we end up with ADP as well. This reaction is one that we had seen a little earlier, where we were looking at energy coupling reactions. So the process of combining glucose and phosphate to form glucose 6-phosphate is endergonic and will require the help of ATP for coupling reactions 1 and 2 to form in reaction 3, which is a reaction that takes place as the first step of glycolysis. I'm going to show you a little video of the hexokinase enzyme and its activity. So hopefully that will help you review how enzymes work. And now this is the specific case with this chemical reaction. So this is the enzyme hexokinase. Here we see the substrates, glucose and ATP. And so glucose and ATP are going to join at the active site of the enzyme, and a phosphate is going to be transferred to glucose. Okay, here you will see the phosphate being transferred to glucose, so pay attention. On the right side, ATP, so you'll see that movement. Phosphate is transferred. Okay, let me replay just this part. Went a little fast. Phosphate transferred. Okay, let me play it one more time. Phosphate is transferred. So then the 2 products can exit the active site and then diffuse away. So hopefully that was useful with the nice visuals. Now, we have said that in metabolic pathways, some steps are irreversible. And so how can that be beneficial? In this case, this reaction is irreversible. So you have to think here that for glucose to be converted to glucose 6-phosphate, it must first enter the cell, right? So this reaction occurs in the cytosol from glucose to glucose 6-phosphate. And so glucose first enters the cell via a transporter that we call GLUT. And that transporter allows glucose to move in either direction. So glucose is actually going to move by passive transport down its concentration gradient. But if we want the reactions to take place, we need to have our glucose moving into the cell. So what we do is we convert glucose to glucose 6-phosphate, and therefore glucose 6-phosphate cannot cross the GLUT transporter. So I guess I could write like that. And therefore, we're going to decrease the concentration of glucose into the cell, and that will allow more glucose to flow into the cell down its concentration gradient. And then we're going to be able to move forward with glucose 6-phosphate through the next reactions. Okay, so this information is written here as well. So in general, irreversible steps are important in metabolic pathways. So we've mentioned that as a general comment before. So let's look further at some other reactions. So the second reaction in the process is called isomerization. We're forming isomers. And this is a reversible reaction where we can go from glucose 6-phosphate to fructose 6-phosphate. By the way, when we see that number 6 is because it indicates where it is attached, to which carbon it is attached. And so what we do in this process is just to shift one of the carbonyl group, okay, with the oxygen here, from one carbon, attached to one carbon, and attach it to another carbon instead. That's all. So we form isomers. If we look at reaction number 3, this is a phosphorylation reaction. Phosphorylation of the fructose 6-phosphate substrate. We're going to use one ATP molecule here, and we're going to transfer our phosphate from the ATP to the substrate. And we have ADP as a result as well. And you can see now that the resulting product, fructose 1,6-bisphosphate. So 1,6 because the first phosphate is attached to the first carbon, and the other phosphate is attached to carbon number 6. Okay, so what we did is we added a phosphate group here. So in the preparatory phase of glycolysis, which consisted of 5 chemical reactions, we ended with forming 2 molecules of glyceraldehyde 3-phosphate. And so in the payoff phase of glycolysis, so the last 5 chemical reactions of the pathway, we're going to convert this molecule, glyceraldehyde 3-phosphate, to pyruvate. It's going to be oxidized further. And in that process, we're going to make 4 ATP molecules. Now, we had invested 2 ATP molecules in the preparatory phase, so our net gain will be of 2 ATP molecules. And we're also going to reduce 2 NAD plus molecules to NADH. So we're forming 2 NADH molecules. These are electron carriers that are going to be used later on. But now we form pyruvate. And so what do we do with pyruvate next? Well, it depends if there's oxygen in our cell. If there is oxygen, we're going to send that pyruvate to the mitochondrion to carry the following steps, the linking steps, stage 2, and then stage 3. If there is no oxygen, well, we're going to discuss that later. Now, before we move on to the linking step, I would like to go over some interesting reactions here of the payoff phase. Again, not to overwhelm you with details, but rather to show that it's possible to understand. So the 2 chemical reactions we're going to look at are number 6 as well as number 10. Reaction number 6 is a redox reaction, so oxidation reduction. And we start with glyceraldehyde 3-phosphate with an inorganic phosphate as well. and adding NAD plus. And what we're going to do is we're going to oxidize our glyceraldehyde 3-phosphate. At the same time, we're going to reduce our NAD plus to NADH. What that means is that we're going to add 2 electrons as well as 1 hydrogen ion, and the other hydrogen ion will be released into solution. This reaction is actually also a phosphorylation reaction because we're phosphorylating, we're adding a phosphate to our carbon-based molecule. The last reaction in the glycolysis pathway is an example of substrate-level phosphorylation. So with the help of the enzyme pyruvate kinase, we're going to phosphorylate ADP to ATP. We're adding 1 phosphate forming ATP. And therefore, we're losing a phosphate from our carbon-based molecule as well. We end up with the molecule pyruvate, which then is going to feed into the next step in the process. Our next step, called the linking step, will allow us to transport pyruvate from the cytosol to the mitochondrion and convert pyruvate to acetyl-CoA. Now, in the cytosol, this is where glycolysis, the pathway of glycolysis, was taking place. And the end product of glycolysis was pyruvate. In the mitochondrion, the linking step is going to take place, the reactions involved, as well as the citric acid cycle. That's our next stage in aerobic respiration. So citric acid cycle will also occur there. So that brings us to discuss a little bit the anatomy of the mitochondrion. So what we see is that we have an outer membrane, and then a tiny space, and then an inner membrane, which is very much folded, and those folds we call cristae. And then internal to the inner membrane, we have that space of the mitochondria, the main part, which we call the matrix. Now, we're going to use an enzyme complex. We call it the pyruvate dehydrogenase complex. So what is a complex? It contains several enzymes that work together to carry out different steps in a chemical reaction or in a series of chemical reactions. So you can imagine that complex contains 3 enzymes, each of them responsible for 1 step. So enzyme 1, for example, is going to allow us to remove 1 CO2 from our pyruvate. That's we call decarboxylation. So you can see in the pyruvate molecule, the carbon and the 2 oxygens will be removed. So now our pyruvate loses a carbon. So it started with 3 carbons, and it will become a 2-carbon molecule, eventually forming what we call an acetyl group. Okay, so that's where we see the acetyl group here. We're also going to carry a redox reaction, oxidation reduction. So pyruvate is going to be oxidized. And this is carried by another enzyme, enzyme 3. And we're going to transfer some energy by reducing NAD plus to NADH. Finally, we're going to add to the acetyl group another molecule that we call coenzyme A, so that we will form Acetyl-CoA. Okay, and that's carried by the other enzyme of the complex. And then acetyl-CoA, which now contains only 2 carbons, will enter the Krebs cycle or the citric acid cycle.